Have you ever had that, like, nagging feeling that you only know maybe 1% of the story?
Oh, absolutely. It's like walking into a movie theater right at the climax.
Right. You sit down and you have zero idea how any of the characters got there or, why everything on screen is suddenly exploding.
Exactly. And for decades, that is pretty much exactly where the field of biology has been sitting.
Yeah. I mean, we mapped the human genome, we threw massive parades to celebrate, and then we just spent the next 20 years obsessing over the 1 to 2% of that DNA that actually codes for proteins.
Yeah, completely ignoring the rest.
Right. But what about the other 98%?
The so-called junk DNA, which, spoiler alert, is absolutely not junk.
Not even a little bit.
No. It is this massive, intensely complex regulatory network, and it's operating entirely in the shadows. We've known it was there, but it's just been incredibly difficult to actually observe what it's doing in real time.
And today, we are shining a massive spotlight on that genetic dark matter. We are jumping into a really landmark May 2026 paper from Nature Methods.
Yeah, the title is Unraveling lncRNA Diversity at a Single-Cell Resolution and in a Spatial Context Across Different Cancer Types.
It's a mouthful, but the mission for this deep dive is to explore how scientists are finally illuminating this dark matter in cancer. They discovered almost 95,000 completely unannotated, basically hidden pieces of genetic code. And we're going to break down what this actually means for the future of medicine.
It's going to be a fascinating one.
Okay, so let's unpack this before we get into all the crazy spatial technology they used. What exactly is a lncRNA?
So lncRNA stands for long non-coding RNA.
Okay.
And if we break that down, long just means it's over 200 nucleotides in length.
Got it. So it's not some tiny fragment, it's a substantial, structurally complex molecule.
Exactly. And non-coding means it does not contain the instructions to build a protein.
Right, because the traditional biology dogma we all learned in high school was pretty simple, right? DNA makes RNA and RNA makes protein.
Right, but lncRNAs stop at that RNA stage. They don't build anything physical. They do their actual work directly as RNA molecules.
I'm trying to picture this. So if your DNA is like the master blueprint locked in the bank vault.
Sure.
And your standard messenger RNA, the mRNA, those are the specific work orders sent down to the factory floor to build the machines, the proteins.
Right.
Where do these lncRNAs fit in? Are they just sort of wandering around the factory?
Think of them as the middle managers and the foremen of that factory.
Okay, the middle manager.
Yeah, they don't actually build the machines themselves, but they are constantly walking the factory floor. They control when, where, and how the entire production operates.
So they have a lot of power.
Oh, absolutely. They can shut down whole assembly lines, speed them up, or redirect resources to different parts of the cell based on, what the environment demands.
That makes total sense. But if they are so vital to managing the factory, why has it been so hard for us to see them clearly until now?
Well, historically, scientists used a technique called bulk sequencing to look at RNA in tumors. And the major problem with bulk sequencing is that it completely destroys the physical context of the tissue.
Wait, so we were just taking a tumor biopsy and basically mashing it all up?
Basically, yeah. It's like taking a beautifully arranged fruit bowl and just throwing the whole thing into a blender.
Oh, wow. So you get a biological smoothie.
Exactly. You might taste strawberry and banana in that smoothie, meaning certain genes are active somewhere in that tumor, but you have no idea if the strawberry was sitting next to the banana originally.
Or which specific cell they even came from.
Right. You lose all the spatial architecture.
So how do we unblend this smoothie? How did the researchers in this specific paper actually find these 95,000 new managers?
They leveraged two massive breakthrough technologies: single-cell RNA sequencing and spatial transcriptomics. Specifically, they used a system called 10x Visium.
Okay, so what does that actually look like in the lab?
Imagine being able to look at that intact fruit bowl, completely unblended, and knowing the exact molecular makeup of every single piece of fruit, exactly where it sits.
That is wild.
They use these specialized glass slides covered in millions of microscopic barcoded probes.
Barcoded probes, like scanning items at a grocery store checkout.
Very similar, yeah. They place a microscopic slice of intact tumor tissue directly onto this slide.
Okay.
And when the RNA is extracted from the cells, it gets snagged by those probes and automatically tagged with a spatial barcode.
Oh, I see. So this allows the researchers to map exactly which RNA molecule came from which exact microscopic coordinate in the tissue.
Exactly. They literally create a high-definition, two-dimensional map of the factory floor showing exactly where every single middle manager is standing.
And they did this at a massive scale for this paper, right?
Oh, yeah. They looked at 13 different cancer types: melanoma, breast cancer, head and neck, colorectal, kidney. It was a huge data set.
And to make sense of all that spatial data, they ran it through this highly advanced computational pipeline. They called it tar-cRNA-seq.
Right.
And it uses something called a hidden Markov model. You're going to have to explain that one to me.
Right.
What is the hidden Markov model?
Sure. Think of it like a highly advanced text prediction algorithm, but applied to biology. It scans the genome in tiny 50 base pair chunks.
Okay, tiny chunks.
Yeah, and it's looking for statistical probabilities of what they call hidden states. Basically, it's looking for complex patterns that suggest active construction is happening.
Meaning RNA is actively being transcribed.
Exactly. Even if our standard biology textbooks say that specific area of the genome should just be an empty, inactive parking lot.
And this algorithm, it hit the absolute jackpot. It found over 219,000 potential lncRNAs. They termed them QTARs, cancer-associated, unannotated, transcriptionally active regions.
That's right.
But the part that really blew my mind was when they cross-referenced those 219,000 findings with 5 of our massive existing genetic databases.
Yeah, the validation phase.
They realized that almost 95,000 of these were completely unannotated. They were ghost transcripts, like nobody had ever documented them before.
Brand new discoveries operating completely in the dark.
But hold on, I have to play the skeptic here and ask the obvious question. If there are almost 100,000 of these hidden managers pulling the strings in cancer, how on earth did the entire global scientific community miss them until 2026? Are they just invisible?
It's a vital question. And no, they aren't invisible, but they are incredibly low abundance, and they're highly specific to tiny, localized neighborhoods within a tumor.
Oh, so think back to the smoothie analogy.
Exactly. If a specific lncRNA is only active in a tiny subpopulation of tumor cells at the very invasive edge of the cancer, the cell's actually pushing into the healthy tissue.
Right.
Blending the whole tumor dilutes that specific signal down to zero. It just looks like random background noise in the smoothie.
But by preserving the spatial architecture, the researchers could finally see these intense, localized spikes of activity.
Oh, yeah.
Okay, so let's do a quick recap here. We know that 98% of our genome is non-coding. And by moving away from blending tumors and instead using single-cell and spatial mapping, scientists found around 95,000 completely new lncRNAs across 13 different cancers.
That is the big picture, yes.
So if you're listening, think about your own body right now. If you have this vast, hidden regulatory network operating in the background of your cells, how might it be adapting right now without you even knowing? It really makes you wonder.
It's a profound thought. But, finding a signal in a computer algorithm, no matter how advanced that algorithm is, that is only the first step. Biology is messy.
Very messy.
The scientific method demands rigorous physical validation.
Right. Which brings us to the skeptic's next big hurdle. Proving this isn't just an artifact. How do we actually know the computer algorithm didn't just hallucinate a bunch of background genetic noise and call it a breakthrough discovery?
That was the exact challenge the researchers faced, and it was largely due to a mechanical limitation of the standard spatial technology they initially used.
The 10x Visium.
Right. Standard spatial transcriptomics usually relies on something called poly-A capture.
Poly-A capture, okay. What does that actually look like?
Well, most mature RNA molecules have a tail made of adenine nucleotides, a poly-A tail. So the sequencing machine basically throws out a chemical hook that only grabs that tail.
Oh, I see. So it's snagging the very end of the molecule.
Exactly. It grabs the tail, and then it only sequences a short, fragmented piece of that end.
I'm trying to visualize that. It's like trying to identify a massive freight train just by looking at the caboose.
That's a great way to put it. You know a train is there, but you have no idea how long it is, what kind of engine it has, or if the cars ahead are carrying coal or passengers or hazardous waste.
So to prove these were full functional RNA molecules like entire trains, they had to upgrade their cameras.
Exactly. They had to capture the whole train, so they turned to long-read spatial sequencing, specifically Oxford Nanopore and PacBio Maya-Seq technologies.
And how do those work differently?
Instead of fragmenting the RNA or just grabbing the tail, these long-read machines literally pull the entire unbroken RNA molecule through a microscopic electrically charged pore.
Wait, they pull it through a pore?
Yeah.
Like threading a needle?
Basically, yeah. And as the molecule passes through, each individual building block of the RNA creates a distinct electrical signature, so they can read the sequence from end to end.
Wow. So they captured the whole train. And in the paper, they talk about how they applied this to a sample of head and neck cancer. The nanopore sequencing physically confirmed nearly 1,000 of these new QTARs in that one sample alone.
Right.
But they didn't just rely on the sequencing, did they? They actually wanted to physically see them inside the cells.
Yeah, seeing is believing. So they used highly advanced in situ imaging tools, things like Xenium, Stomix, and the Takara Bioseeker.
Which act like microscopic glow-in-the-dark GPS trackers, right?
Exactly. They designed chemical probes specifically engineered to physically latch onto these brand new RNAs right inside the intact tissue. Then, using advanced fluorescent imaging, they literally took high-definition pictures of these unannotated lncRNAs.
And they were sitting exactly where the algorithm predicted they would be.
Right inside the specific tumor cells.
That is incredible.
And what's fascinating here is that the evidence goes far beyond just proving they exist physically, because, a random piece of genetic debris wouldn't have any real structure.
It could just be floppy nonsense.
Right. But researchers calculated the thermodynamics of these sequences and found they fold into highly stable, intricate, three-dimensional shapes.
Oh, wow.
Yeah, they verified they have perfectly placed splice sites, a clear promoter at the starting end, and a proper tail at the finish line.
So to put this in perspective for anyone listening, we aren't just trusting a computer model that spit out some data. The researchers checked the whole train from end to end. They took glowing physical photographs of these molecules inside the tumor cells, and they proved they fold into genuine, beautifully structured shapes.
It takes layers of entirely different technologies to establish a single biological truth.
Okay, time for another quick recap. To prove the computer didn't make a mistake, scientists used long-read tech to sequence the whole RNA molecule and physical imaging to see them directly inside tumor cells.
Spot on.
So when you hear about a breakthrough in the news, do you ever stop to ask what kind of evidence actually backs it up? Notice how all these different layers of technology are required here to establish one single truth. It's not just one experiment.
No, it's a huge convergence of data. And establishing that truth leads us to the most vital question of the whole study.
Right. We know they exist. We know they have structure. We know exactly where they live in the tumor. But what are they actually doing? How are they driving the cancer?
Right. What is their motive? Which brings us to the concept of guilt by association.
Guilt by association. Because if you find an entirely new, unannotated piece of genetic code, how do you even figure out what its job is?
You look at its neighbors. The researchers employed a statistical method called bivariate Moran's I. It's a way to find spatial autocorrelation.
Can you break down how that works in practice? Like, give me an analogy.
Sure. Imagine a bank robbery. You don't know who the getaway driver is. You have no physical description of them. But if you analyze millions of data points and you notice that a specific unknown cell phone always pings at the exact same GPS coordinates at the exact same microscopic moment as a known famous bank robber.
Oh, that's spatial autocorrelation.
Exactly. If a specific unknown lncRNA constantly shows up in the exact same microscopic spot as a known dangerous cancer-driving gene, we can infer they are working together. You found your accomplice without ever seeing their face.
Okay, here's where it gets really interesting. Let's look at how they applied that in a specific case study from the paper. In that head and neck cancer sample, they used this spatial math and found a brand new lncRNA. It was QTAR 213507.
Right.
And it had massive spatial autocorrelation with a gene called FGFR3 on chromosome 4. And FGFR3 is a very well-known bad actor, is heavily upregulated in these cancers, driving massive tumor growth.
And the plot thickens even more there because this specific new lncRNA is also located right next to another gene called 10M3.
Okay, and why does 10M3 matter?
Because that specific gene is a well-known integration site for the human papillomavirus, or HPV.
Oh, wow.
Yeah, HPV insertion at this specific site is a primary driver of certain throat and mouth cancers.
So, using your bank robbery analogy, the 10M3 gene is basically a compromised security door at the bank.
Yeah.
HPV is the burglar slipping through that broken door. And our new discovery, this unannotated lncRNA.
It's the getaway driver.
Right. It's parked right next to the door, highly coordinated with the other cancer genes, waiting to help the tumor evade the immune system and grow. They're operating as a unified crew.
That's a very vivid way to put it, but yes, they are part of the exact same regulatory network. And let's look at a second case study from the paper, this time in breast cancer.
Okay.
The researchers used machine learning to predict what proteins a specific new RNA QTAR 215705 might interact with.
QTAR 215705.
Okay. The model predicted this new RNA acts like a hand that holds a very specific key protein. It's an RNA-binding protein called HER, or ELAVL1.
Okay, so a hand holding a key. What does the key unlock?
That key protein regulates a famous master switch gene called BRC5. And BRC5 is essentially the lock on a cell's suicide switch.
A suicide switch, you mean apoptosis?
Exactly. Programs cell death. Normal cells have this mechanism. If a cell becomes damaged or cancerous, it's supposed to flip that switch and destroy itself to protect the body.
But cancer survived by breaking that switch, right?
Yeah.
They refuse to die.
Exactly. BRC5 is the lock keeping that suicide switch from flipping. The key protein opens it, and it appears this entirely new, unannotated lncRNA is the hand turning the key, actively helping the breast cancer cells stay immortal.
And the data they found to back this up is just staggering. They looked at a cohort of 246 actual breast cancer patients.
Right.
And they found that measuring the levels of this specific, newly discovered lncRNA predicted poor patient survival more accurately than measuring the actual protein-coding gene BRC5 did.
That is a paradigm-shifting data point.
It really is.
Think about the implications. A piece of genetic dark matter, something we considered junk and completely ignored until this study, is a better clinical predictor of human mortality than the traditional gene we've been studying for decades.
Okay, let's do a quick recap here. By mapping where lncRNAs live, scientists found they partner with known cancer genes, basically helping tumors grow and evade cell death.
That's the core finding.
So, question for you listening. If a non-coding RNA is a better predictor of cancer survival than a traditional gene, how much of our standard medical testing right now is currently looking in the wrong place? It implies a massive blind spot in diagnostics.
It absolutely does imply that, which perfectly transitions us to the final piece of this puzzle, the actual clinical impact.
Right, because understanding the deep biology and finding these hidden networks is fascinating, but how does this translate into the clinic? How does this help a patient who is fighting for their life today?
Exactly. We know they manage the factory, but what happens when we actually try to attack the cancer with drugs? Do they fight back? They react very dynamically, and they definitely fight back. The researchers looked at patient data across different treatments to see how these lncRNAs behave under pressure.
Give me an example.
For instance, they looked at melanoma patients receiving immunotherapy, specifically anti-PD-1 drugs. These drugs essentially take the brakes off the patient's immune system so it can attack the tumor.
Okay.
They observed that specific lncRNAs associated with pigmentation genes like PMLL and mLNA, they shifted their expression levels significantly based on whether the tumor was responding to the treatment or resisting it.
Oh, so they changed their behavior based on the drug. And they also looked at medulloblastoma, right, which is a really devastating type of brain tumor.
Yes. And to study it, they used something called a PDOX model. Essentially, take a sample of a human brain tumor, and they grow it in the exact same anatomical location inside a mouse's brain.
Wow, so it's a highly realistic environment.
Very realistic. And they treated these tumors with a targeted drug called palbociclib.
And what does palbociclib actually do?
It specifically halts cell division. It just stops the tumor from multiplying. And when they introduced this drug and watched the spatial map, they saw specific, newly discovered lncRNAs completely shut down.
Really?
Yeah. Their activity mirrored the exact behavior of known dangerous oncogenes.
So they aren't just static bystanders. They were actively involved in how the tumor responds to or resists chemotherapy.
Exactly. And if we connect this to the bigger picture, this isn't just about finding new markers so we can give patients a more accurate prognosis. It's about finding entirely new, untapped targets for actual therapies.
Because if a cancer becomes resistant to a traditional chemotherapy drug, and we find out a specific lncRNA manager is the one driving that resistance.
Oh, we can design a therapy to block that specific manager. If you remove the foreman directing the resistance, the factory collapses. It opens up tens of thousands of new therapeutic avenues.
What I absolutely love about this paper is that the researchers didn't just hoard all this incredible data. They didn't lock it behind some patent.
No, they didn't.
To make sure those therapeutic avenues are explored quickly by the global community, they created a massive database called the SPanC-Lnc Atlas. And it's hosted on the AWS cloud, completely open access.
It is essentially a public Google Maps for cancer researchers.
A Google Maps. I love that.
Yeah. Anyone, anywhere in the world can log in, select a specific cancer type, zoom into a microscopic cell population, and see exactly which of these 219,000 lncRNAs are active there.
It shows their predicted structure, their genomic neighbors, everything.
Everything. It utterly democratizes the dark matter.
It's just an incredible resource. So let's recap this final segment. We saw that lncRNAs dynamically change their behavior when tumors are treated with drugs, making them perfect targets for future therapies. And all this spatial data is now completely free in the SPanC-Lnc Atlas.
A huge win for open science.
Absolutely. So listener, with this open source map of cancer's hidden managers now live, what kind of rapid breakthroughs do you think we'll see in the next 5 years? The acceleration is going to be wild.
Oh, it's going to be exponential. We are moving from a blurry, low-resolution understanding of the tumor environment to a high-definition, single-cell spatial reality. It is a phenomenal piece of integrative biology.
It really is. So to bring it all together, we started today with the mystery of the 98% non-coding genome. We saw how cutting-edge spatial tech illuminated nearly 95,000 completely new RNA molecules.
We unblended the smoothie.
You unblended the smoothie. We walked through how scientists proved they physically exist using end-to-end long-read sequencing and glowing fluorescent trackers.
We're capturing the whole train.
Exactly. We explored how they act as accomplices to known cancer genes, and we saw how they dynamically react to life-saving drugs.
It's a completely new frontier for medicine.
It really is. But before we wrap up, I want to leave you with one final provocative thought. This entire massive study, this colossal effort, which uncovered 95,000 new genetic players misbehaving and pulling the strings, it only looked at 13 types of cancer.
Which is just a tiny fraction of human disease.
Exactly. If we just discovered 95,000 new middle managers operating in the shadows of these specific tumors, what on earth is the genetic dark matter doing right now in your completely healthy cells?
It's a staggering thought.
Right. What is it doing in the brain during the onset of Alzheimer's? Or in the tissue of the heart during cardiovascular disease? We haven't just found a new piece of the puzzle. We've discovered that the puzzle itself is 100 times larger and infinitely more complex than we ever thought.
The dark matter is waking up, and we are finally building the tools to see it.
Keep questioning the 99% of the world we can't yet see. Thanks for joining us on this deep dive.